Every day, we are encountering thousands and thousands of pathogens, and it turns out our body's immune system can mount a defense response against them. So how does our immune system does that? One of the component of our immune system is B cell. Against the pathogenic antigens, B cells produce antibodies. Actually, B cells get differentiated into antibody secreting plasma cells. Now, these plasma cells can specifically mount immune response against a pathogenic antigen. So, there are thousands of antigens could be produced by a single pathogen itself. And imagine there are so many pathogens, and our immune system need to be diverse in terms of creating new versions of antibodies such that it can encounter the pathogenic antigens. So this diversity is required for antibody production. Question is, how does this diversity achieved in terms of antibody production, or how does diverse antibodies with different affinity and avidity for a particular antigen is created? And the answer lies in a genetic recombination which happens in the antibody light chain and heavy chain genes. So in this video, we'll talk about VDJ recombination. It turns out we have 51V, 27D, and 6J gene segments. So the simple thing is we don't have one gene segment for heavy chain or the light chain of the antibodies. We have multiple gene segments. Now, what is the importance of having so many different segments? It turns out these several segments could be joined in a sequence. For example, this diversity and J segment can join together by a recombination process known as DJ recombination. Further, the VDJ, these segments could be joined in VDJ recombination process and thereby can create a different transcript depending upon which combinations they are combining, and that can give rise to antibodies heavy chain. Now, in a combinatorial manner, huge diversity could be produced itself in the heavy chain. Forget about the light chain for a moment, but itself in the heavy chain, using a combinatorial code, a plasma cell can produce huge variety of antibodies. Now, we talk about the light chain. Light chain doesn't have the J segment, so doesn't have the diversity segment, but it has the V and J segments, which could be recombining together by VJ recombination to give rise to specific transcript which can form specific light chain segments. Now, not only B cells or B cell receptors or antibodies undergoes this kind of recombination to produce diversity. It turns out that B cell receptor and T cell receptor all undergoes this kind of recombination to produce the B cell receptor or T cell receptor. Now, VDJ recombination is dependent upon two different components. First, the recombination signal sequence present in the gene segments. Second, the recombinase enzyme that helps in recombination. So it turns out after every gene segments, there are small signal sequences. These signal sequences are either known as one-turn sequences or two-turn sequences. The one-turn sequences have 12 base pairs spacer in between them, whereas two-turn has 23 base pair spacers between them. And there is a joining rule between these gene segments and these recombination signal sequences. The rule says that opposite turns can join together. For example, a one-turn sequence can join with a two-turn sequence, but the similar ones cannot join with each other. This ensures that the gene segments recombine in a proper order, and also the internal recombination between itself, let's say the diversity segment, or itself between the joining segments cannot happen. So the intra-segmental recombination cannot happen by following this rule. So which is a clever way of designing this recombination system that evolved throughout time. Now, let's talk about the enzymes which are necessary for this recombination process. As expected, since it's a recombination process, there should be specific recombinase. So there are two recombinase of RAG1, which is known as recombinase associated gene, their product give rise to RAG1 and RAG2 recombinases, which are most important recombinases for VDJ recombination. Apart from that, terminal deoxynucleotide transferase or TDT is also involved in this process. Now, B cell development starts in the bone marrow, right? After its development in the bone marrow, it moves to the lymph node where it gets matured and learn how to encounter pathogens. Now, inside the bone marrow, there are vascular niche and endosteal niche where several stages of the B cells could be found. Let us look at which stage of the B cell this VDJ recombination is taking place. So, in pre-pro B cell stage, the pre-pro B cell gets attached to the bone marrow stromatal cell, and one important interaction is SCF and c-KIT, and also it expresses several chemokine receptors such as CXCL12 receptor. So all these signals give rise to DJ recombination at this particular point of time of the heavy chain. Later, when it progresses to early pro B cell stage, then interleukin derived from the stromatal cells to give rise to specific signaling that helps in total VDJ recombination process. Other than these external signals, the external signals impinge upon internal signals onto internal transcription factors, which helps in this process, maybe by producing the recombinases gene or many other aspects of the recombination. So we'll look at what happens when IL-7 mediated signaling comes and how does the immature B cell read that signal. So here is IL-7. IL-7 give rise to JAK-STAT mediated signaling, which allow the activation of several transcription factors like EPF1, PAX5, etc. All of these help in VDJ recombination or several aspects of VDJ recombination. So let's just look quickly at the mechanism of VDJ recombination. So the first step of the recombination is the enzymes RAG1 and RAG2 should recognize the proper recombination signal sequence on the VDJ gene segment. So here you understand there is, let's say we take a simple example of a light chain gene segments where we have V and J. So here our recombination associated proteins RAG1 and RAG2 is recognizing the signal sequences. After that, these enzymes use a DNA looping mechanism by which this 1-turn and 2-turn RSS are paired inside the active site of these enzymes. And there is a cleavage of these one single strand by RAG1 and RAG2. After that, these cleavage sequences form hairpins with each other. And this is followed by a double-stranded DNA break by RAG1 and RAG2. So when the double-stranded DNA break occurs, it needs to be joined together. And that joining process happens by NHEJ mediated mechanism, where Q70 or Q80 binds to it, followed by the binding of Artemis, DNAPK, and XRCC4, etc. enzymes of all the NHEJ mechanism, which give rise to joining of these gene segments. And now the mRNA would be produced from this gene segment, which would be later translated to give rise to the light chain. Now we talk about a little bit how this diversity could arise in terms of after VDJ recombination, or what are the mechanism of producing antibody diversity. So it turns out multiple germline gene segments are there that we looked at. So there is at least 51 V, 27 D, and 6 J chains. That means possible combinations that could be achieved by using a combination is at least more than rough about 8,000. Apart from that, there is a junctional flexibility. That means whenever there is a double-stranded break, when the segments join together, there is a flexibility in this joining process. Not only there is a junctional flexibility, whenever the double-stranded breaks happen, there could be new nucleotide addition to it, which accounts for diversity. And after all these things, there is a process known as somatic hypermutation, which creates even more diverse antibodies. So I hope you enjoyed this video. If you like